Hi, this is Michael Edwards. I'm an assistant professor of medicine at the University of Colorado of Denver, specializing in bioinformatics. This is part one of my tutorial looking at using David. David is the bioinformatics resource developed by the National Institute of Allergy and Infectious Disease, as well as NIH. This is probably the most popular and most used free bioinformatic resource available. So this first part will go into how do we analyze or how do we upload our data set or our gene list into David. To start with, you can just Google this website. It's pretty easy to get to. This is the home page. So what I'm going to do is hit Start Analysis, which is up in the left corner here, which will bring you to this page. We want to upload our list. So I'm going to select this tab, Upload. Right here, down here is this window where you can paste your gene list or you can upload an Excel file. For this demonstration, we're going to upload a gene list. It's very easy. I'm going to go to my original list. This was developed by looking at the differences between aggressive and non-aggressive types of B-cell lymphoma. This was in the AACR workshop. This was an RNA-seq study looking at this, and at an FDR of less than 0.5 or a false discovery rate of less than 5%, we were ending up with 748 transcripts, and they are given here. So I'm going to go to this left column, which gives the official gene names for our list, and I'm just going to go all the way to the bottom and select all of these. Edit, copy. Now I'm going to go back to the David website and just paste our list here. Okay. We're going to go down. Again, this is the text file option. We want to select what identifiers these are. As I said, these are gene symbols, so we're going to select that. Official gene symbol here. You got to tell the software what type of or what the identifier is. And then we're also going to select gene list. So this gene list is basically, this option is the genes that are different between your two conditions or whatever conditions you're looking at. The background option tells the software how many genes you had to choose from. Since we did an RNA-seq study, we're actually surveying all the genes in the genome. So our background list is just going to be a species, and you'll see where I can select that option. So I'm going to hit Submit List down here on the left. Okay, this is where it tells us, do you want what type of background you would like to use. So I'm going to hit OK. Which brings us to this tab, the list. I'm going to select Homo sapiens here. These were done on human cells. Select species. And you'll see it pop up over here, Homo sapiens. I'm also going to change our list manager or our list name. I'm going to rename, and we're going to put the two cell types we're looking at. One of those was the activated B cell type, which is the more aggressive version of this B cell lymphoma, versus the germinal center B cell like, which represents our less aggressive form of this type of cancer. I'm going to hit OK. And now we're just going to, and as we do that, now we have the options, the analysis options for our gene list are available here. In the second tutorial that I'm going to look at, first what I'm going to do is how can we explore this list on the gene level. So we're going to look at gene functional classification tools, how to take these, or what these genes mean, what are some of its relationships, and then look at gene ID conversion tool. How can I take my list and convert it into another identifier that I can use with other software? So on to the second part of the David tutorial. Thank you.